Okay, so welcome to this next video in which we are discussing the adaptive immune system and the immunosuppressant drugs. Okay, so we are currently looking at immunosuppressant drugs which work by inhibiting T-cell activation. Okay, so we've now looked at the way that signal 2 and signal 3, signal 1 and signal 2 affect the T-cell. They cause a rise in calcium in the cytoplasm. This calcium activates calmodulin and forms calcium-calmodulin complexes, which then activate calcineurin enzymes, which remove the phosphate groups from the nuclear factor of activated T-cells. This allows the nuclear factor of activated T-cells to move from the cytoplasm into the nucleus, where it increases the expression of interleukin-2 and the interleukin-2 receptor alpha. The interleukin-2 is then secreted from the cell and will bind to the interleukin-2 receptor and cause signal 3. Okay, so we're now in a position to look at 3 drugs which work by stopping T-cell activation. Okay, so let's start with the first one. Okay, now where shall I put this? I'll put this, now I'll go over onto the other page. Okay, so the first drug is a drug known as cyclosporin, and this is a really famous immunosuppressant drug. So, what does cyclosporin do? And it's also sometimes known as cyclosporin A, but often just as cyclosporin. So what does cyclosporin do? Well, basically, cyclosporin is going to come in. So let's say this is our cyclosporin drug. So I'll give it a colour. So we'll colour it in green here. It's going to come into the cells. Okay, so you'll take it and it will go into these naive CD4 positive and naive CD8 positive T cells. And in the cytoplasm of these cells, it will bind to a protein, okay, known as cyclophilin, okay? So this is the protein cyclophilin, which is in the cytoplasm of the CD4 positive and CD8 positive naive T cells. Okay, and now what it will cause is it will cause this cyclophilin protein to then go and inhibit calcineurin. So this complex of cyclosporin with cyclophilin is going to inhibit the calcineurin enzyme. So even if the calcineurin enzyme gets calcium-calmodulin complexes binding to it, this cyclosporin and cyclophilin dimer here will stop the calcineurin enzyme from becoming active. Okay, so even if the calcium goes up in the cytoplasm of the naive CD8 positive or CD4 positive T lymphocyte, which indicates that it's received signal 1 and signal 2, then that's not going to cause the activation of calcineurin if cyclosporin A is bound to cyclophilin and this dimer here is inhibiting the calcineurin enzyme over here. Okay, and therefore if you don't get activation of calcineurin, then you're not going to get the removal of the phosphate groups from the serine-rich region of the nuclear factor of activated T cells, and therefore you're not going to get the activation of the nuclear factor of activated T cells, and therefore you're not going to get the production of interleukin-2 and the interleukin-2 receptor alpha, and hence you're not going to get signal 3, so the T cell will not undergo complete activation. Okay, so that's how cyclosporin works. Now there's another drug known as tacrolimus, which works in a very similar way by inhibiting calcineurin. Okay, so it's tacrolimus, and it's also got another name that's worth knowing, which is FK506. So before it was actually used clinically, this was its experimental name as an experimental drug. So drugs which aren't used clinically have names that look like this, where you've just got letters and symbols, basically. When they're used clinically, they're given a more catchy name, like tacrolimus, okay? But it's worth knowing that tacrolimus is also known as FK506, because this drug is going to bind to a protein that has been named after this drug, okay? So the protein was named because it's the target of this tacrolimus drug, and the name of the protein was named after the FK506 name for this drug rather than the tacrolimus name. Okay, so let's have FK506 or tacrolimus in red here, and it's going to bind to a protein, again in the cytoplasm of the CD4 positive or CD8 positive naive T cells, and this protein is known as FKBP12, which stands for FK506. Okay, so that's the FK portion. So FK506 binding protein, that's the BP, and then binding protein 12. So the FK506, or the tacrolimus, is going to bind to the FK506 binding protein 12 within the cytoplasm of the naive CD4 positive or CD8 positive T cells. And this complex, this FK506 FKBP12 complex, is going to then inhibit calcineurin and prevent its activation. So again, even if this naive CD4 positive or CD8 positive T cell gets signal 1 and signal 2, which cause calcium to go up, that will then lead to the calcium binding to the calmodulin, and you'll get calcium-calmodulin complexes going up within the cytoplasm. And that now will not lead to the activation of calcineurin. So calcineurin remains inactive even though the calcium-calmodulin complexes in the cytoplasm go up. Okay, and if calcineurin remains inactive, then you're not going to get the removal of the phosphate groups from the serine residues of the serine-rich region of the nuclear factor of activated T cells, and therefore the nuclear factor of activated T cells is going to remain in the cytoplasm inactive, and you're not going to get the increased expression of interleukin-2 and interleukin-2 receptor alpha, and therefore you're not going to get that autocrine signal which delivers signal 3, which is needed for the T cells to complete the activation process. So the T cells aren't going to activate, and therefore again you're going to kill all 3 branches of the adaptive immune system, because all of them required the activation of some form of T cell, whether it was a CD4 positive T cell or a CD8 positive T cell. Okay, so one final drug that's going to work by inhibiting the pathways that we've looked at so far is a drug known as basiliximab, and this I can draw up here. So basiliximab is a drug which binds to the interleukin-2 receptor here. Okay, so once you form the interleukin-2 receptor, we have a monoclonal antibody basically that can bind to the interleukin-2 receptor. Okay, and this is known as basiliximab. Okay, basiliximab, and it's a monoclonal antibody. So here is its antibody structure, and I'm sorry it's squashed in here. Okay, so we have 2 heavy chains, 2 light chains, and its target, the molecule that it binds to, the protein it binds to, is this interleukin-2 receptor. Now I don't know specifically where it binds to on this interleukin-2 receptor. It binds somewhere on the extracellular domain. So I'm sorry that I've sort of shown it binding to the beta subunit. I don't know if it specifically binds to a portion of the beta subunit. It binds, I think, to the receptor as a whole, basically. So I think you need all 3 of them present for it to actually bind. So it will bind to these interleukin-2 receptors, and then it will block the extracellular domain from the interleukin-2. So interleukin-2 will now not be able to bind to this interleukin-2 receptor, and therefore, even if the cell does produce interleukin-2 and the interleukin-2 receptor alpha, the drug will bind and therefore block the interleukin-2 from being able to bind to the interleukin-2 receptor, and that will stop signal 3 from happening, basically. So you won't get signal 3, and therefore you won't get the full activation of these CD4 positive or CD8 positive naive T cells. Okay, so that drug is basiliximab. Okay, and it's a monoclonal antibody against the interleukin-2 receptor. Okay, so these 3 drugs, cyclosporin A, tacrolimus or FK506, and basiliximab, work by blocking T cell activation, and they stop the induction of signal 3, basically. They stop signal 1 and signal 2, causing signal 3. Okay, now the next drug we're going to look at also works by blocking the activation of T cells, but it works by stopping signal 3. So you do get signal 3, but it's going to stop it, basically. And this is a rather famous drug. Okay, so the drug we're now going to explore is rapamycin, okay, which has a new name now. It's also known as sirolimus. Okay, so rapamycin is going to work by intercepting signal 3, basically. Okay, so let's now discuss signal 3 in more detail, because in order to understand how rapamycin works, we need to discuss what signal 3 actually is. So, let's go back to our interleukin-2 receptor here. So here is the phospholipid bilayer, and it consists of these, and here is the interleukin-2 receptor, which consists of these 3 subunits, the alpha, the beta, and the gamma subunit here. Okay, now, what happens when interleukin-2 actually binds to the interleukin-2 receptor? So here is the interleukin-2 binding to the interleukin-2 receptor. Well, basically, you have certain enzymes bound to the cytoplasmic domains of these proteins. Okay, so you have an enzyme bound to the beta subunit of the interleukin-2 receptor, and you also have an enzyme bound to the cytoplasmic domain of the gamma subunit of the interleukin-2 receptor. Okay, so let me add some colour on here to make it look simpler. So here is an enzyme bound to the cytoplasmic domain of the interleukin-2 receptor beta subunit, and here is an enzyme bound to the cytoplasmic domain of the interleukin-2 receptor gamma subunit. Okay, so what are the names of these enzymes? So, this one in turquoise here is what's known as JAK1, okay, which stands for Janus Kinase 1. Okay, so JA is for Janus, and then the K is for kinase, and then it's 1. Okay, this next one that's bound to the gamma subunit is JAK3. Okay, so again, Janus Kinase 3 is what that stands for. Okay, and both of these enzymes are what are known as tyrosine kinase enzymes. They stick phosphate groups onto tyrosine residues. So, I discussed what a serine-threonine kinase was so that we could discuss what a serine-threonine phosphatase was, and we found an example in calcineurin. So I think it would be unfair of me now not to discuss what a tyrosine kinase is. So, let's firstly discuss the structure of the amino acid tyrosine. So again, I'll show this structure as though it's a residue within a protein. So here's the amino group that will be linked to the carboxylic acid group of the next amino acid along, and here's the carboxylic acid group which will be linked to another amino group of the next amino acid along in this direction. Okay, then we have the alpha carbon with a hydrogen off it in the middle, and then the R group of tyrosine is that you have a methylene group like so, and then with a benzene ring coming off this methylene group, so here is a benzene ring, and then an alcohol group coming off that benzene ring. So this is the structure of tyrosine. Okay, so again, it has this alcohol group pointing off the side, and we can bind a phosphate group to this alcohol group in the same way as we did before with serine and threonine. Okay, so we can form a phosphoester link. Now these JAK1 and JAK3, those enzymes add phosphate groups onto tyrosine residues. So they are going to phosphorylate tyrosine residues in proteins. Now, when the interleukin-2 binds to the interleukin-2 receptor, it causes the activation of these Janus Kinase 1 enzyme and Janus Kinase 3 enzymes. And they now stick phosphate groups onto the tyrosine residues in the interleukin-2 receptor beta subunit. So, this interleukin-2 receptor beta subunit has a whole bunch of tyrosine residues. So where shall I draw these? I'll have to put them like as a little square here. So you have a whole bunch of tyrosine residues And I'll just denote this little green square to denote the fact that that is an area that has a lot of tyrosine residues in. Okay, so there is a lot of tyrosine residues in this little green square here. And basically what's now going to happen is that these activated JAK1 and JAK3 enzymes are going to stick phosphate groups onto the tyrosine residues on this interleukin-2 receptor beta subunit's intracellular aspect. Okay, right, so then what you have got is a whole bunch of phosphotyrosine residues. So once you've phosphorylated tyrosine, it's then what's known as a phosphotyrosine residue. Now, once you've got phosphotyrosine residues on the cytoplasmic domain of this interleukin-2 receptor beta subunit, that's going to activate a whole bunch of other downstream pathways, which will lead to the cell undergoing firstly differentiation into either a T helper naught cell in the case of CD4 positive naive T cells, or a cytotoxic T cell in the case of CD8 positive naive T cells. And then after that, it's going to lead to the proliferation of these T helper naught cells and cytotoxic T cells. Okay, so that is obviously very, very important for causing the differentiation and proliferation. So if we can block these downstream pathways, then maybe we can stop the proliferation of the T cells, stop the differentiation and the proliferation. So what pathways do these phosphotyrosine residues lead to? Well, it actually leads to a whole host of pathways. Okay, so there are three main pathways that it's going to lead to. It's going to lead to, one, the MAP kinase ERK pathway. Okay, so you'll get the MAP kinase ERK pathway starting off. Okay, so this is a very powerful growth-inducing pathway. It results in the activation of MYC transcription factors, also CFOS, CJUN, heterodimers, very powerful pro-growth transcription factors. So we are going to activate the MAP kinase ERK pathway. Also, you're going to activate from these JAK1 and JAK3s, they're also going to activate what's known as the JAK-STAT pathway. Okay. And finally, it's also going to activate what is known as the PI3 kinase, AKT, and then this one's the most complicated name because it's actually got 3 in its name, mTOR pathway. Okay, now, all 3 of these pathways lead to the differentiation and the growth. Okay, so differentiation and proliferation. So anything to do with differentiation and proliferation is very complicated, so all of these pathways are involved in it. So firstly, they're going to lead to the differentiation and then onwards to the proliferation. Now, the pathway that we are going to look at in detail is we're going to look at this PI3 kinase AKT mTOR pathway. The reason is that rapamycin, or sirolimus, works by interfering with this pathway here, not by interfering with the MAP kinase ERK pathway or the JAK-STAT pathway. So we're going to concentrate on the PI3 kinase AKT mTOR pathway. Okay, so let's look at the PI3 kinase AKT mTOR pathway then now. So, here's a fresh piece of paper. So, the first thing that's going to happen is that these phosphotyrosine residues, okay, which are now exposed on the cytoplasmic domain of the interleukin-2 receptor beta component. So let me draw now the interleukin-2 receptor beta component to have a slightly bigger cytoplasmic domain here. In fact, actually no, let me draw the JAK enzymes back on there. Okay, so I'm just drawing out the interleukin-2 receptor again. Okay, so here is the alpha subunit, here is the beta subunit, here is gamma. Here are those 2 enzymes again, so JAK1 here. Okay, the Janus kinase 1. Okay, and here is JAK3. Okay, and those have, remember, added these phosphate groups onto the tyrosine residues on the cytoplasmic domain of the interleukin-2 receptor beta component. Now, what's going to happen is an enzyme from the cytoplasm is going to come and stick to these phosphotyrosine residues on the cytoplasmic domain of the interleukin-2 receptor beta component. So let me draw this enzyme here. Okay, so let me put it like so. So I'll draw it in the typical way that an enzyme is drawn, with a nice active site here. Okay, and then I'm going to add a little extra bit up here. So we'll put this special little piece up here. Okay, so I'll colour this in blue. Okay, so this special little portion of this enzyme is going to bind to these phosphotyrosine residues on the cytoplasmic domain of the interleukin-2 receptor beta subunit. Okay, and it's what is known as a SH2 domain. Okay, and this stands for Sark homology domain 2. Okay, so Sark homology 2. So this is a SH2 domain, a Sark homology 2 domain. Okay, now it's just a structure within a protein. Okay, so loads of different proteins will have this same structure within them, basically, where it's a similar assortment of amino acids, and it folds into a similar shape, and it's just a subdomain of a protein. So remember, the protein is massive. It has a huge number of different bits. So sticking off up here, it has a subdomain, or just a domain, which is a conserved type of domain. So it's a conserved structure that you see across loads of different proteins that are completely unrelated. And the function of this domain, which is called this SH2 domain, is that it can bind to phosphotyrosine residues. So this enzyme, which is, by the way, known as PI3 kinase, or PI3K. Okay, so let me write the many names for this enzyme. So PI3, and then often people will put kinase. Or in full, its name is phosphoinositide 3 kinase. So that's what the PI stands for. It stands for phosphoinositide and then 3 kinase. Okay, now PI3 kinase is usually within the cytoplasm of the cell. Okay, now when you get these phosphotyrosine residues appearing on the cytoplasmic domain of this interleukin-2 receptor beta subunit, it will migrate. Well, it will not migrate, but it will end up, whenever it comes near that interleukin-2 receptor, which it will do, you know, it's just moving around, and what will happen is it will end up binding to those phosphotyrosine residues on the cytoplasmic domain of the interleukin-2 receptor beta component, and it now gets held right at the phospholipid bilayer. So now it's stuck up here at the phospholipid bilayer. Now it is going to act on a component within the phospholipid bilayer, and I want to stress this. PI3 kinase was already active. It has not been activated by binding to the phosphotyrosine residues. Instead, it has just been relocated. So, putting these phosphotyrosine residues here and binding the SH2 domain of the PI3 kinase to those phosphotyrosine residues does not activate the PI3 kinase. The enzyme was already active, it was just in the wrong place, whereas now it's stuck at the phospholipid bilayer and it's right next to its target, basically, which is in the phospholipid bilayer. So, what is it now going to do? Well, it's going to act on a molecule which is in the phospholipid bilayer, which is known as PIP2. Okay, so I want to go through the structure of this molecule because it's quite important to understand what PI3 kinase is going to do. So, PIP2, or PIP2, its full name is what's known as phosphatidyl inositol 4,5-bisphosphate. Okay, and I just want to draw a little cartoon for what the structure of phosphatidyl inositol 4,5-bisphosphate is. So firstly, let's start by revising the structure of a basic phospholipid within the phospholipid bilayer. So here is the phospholipid bilayer, and let's start with the structure of a basic phospholipid. So we could draw the structure of a phospholipid like so, in cartoon anyway. Okay, so let's talk about the different components of this structure. So here in green, this horizontal line here, this represents glycerol, okay, which is the backbone for the phospholipid. It's also called, or its proper name, is propane 1,2,3-triol. And although propane 1,2,3-triol is a bit of a mouthful, it's useful because it tells you exactly what the glycerol molecule actually is, whereas glycerol doesn't give you a clue as to what it is. Okay, this tells you that it's a free carbon molecule where every single one of the carbons in that carbon molecule has an alcohol group coming off it. Propane 1,2,3-triol. It's a free carbon molecule with alcohol groups coming off every single one of the carbons, or at least just one alcohol group coming off every single carbon. Okay, then let's look at these vertical lines here, which I'm now colouring in orange. These represent the fatty acids, which have been esterified to the first and second alcohol groups of the glycerol molecule. Okay, now again, chemists have a proper name for fatty acids. The proper name for fatty acids is to call them long-chain carboxylic acids. And again, this is nice because it tells you exactly what they are. They are carboxylic acids which have a really long tail, basically. And then you have just esterified them onto the first and the second alcohol groups on this glycerol molecule. Okay, and that leaves one alcohol group now on the third carbon. So what have we added on to this third alcohol group over here? Well, we've added on a phosphate group here. So this little ball represents a phosphate group. Okay, so again, you have forged a phosphate ester link between a phosphate group and the alcohol group in the third position of the glycerol molecule. Okay, so this is a phosphate group. Now the whole structure here is now what is known as a phospholipid, okay? Or an old name for a phospholipid is to call a phospholipid a phosphatidate molecule, okay? And although you'll never hear anyone refer to a pure phospholipid as a phosphatidate molecule, when we refer to modified phospholipids, we use phosphatidate in the naming of them all the time. So look here, phosphatidyl inositol. That is referring to one of these phosphatidate molecules. Okay, so this is a normal phospholipid, but in the phospholipid bilayer, you don't just have normal phospholipids, you have modified phospholipids as well, and this is what PIP2 is. It's a modified phospholipid. So let's take this phospholipid and transform it into this modified phosphatidyl inositol 4,5-bisphosphate. So the first thing is that we have to add on an inositol ring. Okay, so we're going to add this onto the phosphate group here that comes off the third alcohol group of the glycerol molecule. Okay, so what is an inositol ring? So let's just discuss over here what an inositol ring actually is. So, an inositol ring is a 6-membered carbon ring, and I'll draw a skeletal formulae here. So remember, skeletal formulae, we don't show carbon atoms, we show them implicitly as corners, and we don't show hydrogen atoms coming off carbon atoms, okay, but we show everything else. So this is a 6-membered carbon ring where you have 6 carbons here, and all of the bonds within the ring are single bonds. Then, off every single carbon in this ring, you have an alcohol group coming off. So here are 3, here's the 4th alcohol group, 5th is over here, and then finally 6th over here. Okay, and that then leaves every single one of these carbons with 3 bonds. So it all has, each one of them has a single bond left. And again, this is a skeletal structure, so if there's a bond missing off a carbon atom, you assume implicitly that that is a hydrogen atom coming off the carbon. Okay, so every carbon in this 6-membered ring has an alcohol group coming off it and a hydrogen coming off it. So it's cyclohexane, where every carbon has an alcohol group coming off it effectively. Okay, so we'll just denote this as a hexagon, which I'll colour in blue here. So this hexagon represents the inositol ring. And you have taken one of the alcohol groups of this inositol ring and formed a phosphate ester link with the other alcohol group of the phosphate group, and that's linked to this inositol onto this phosphatidyl group, as it's called. So this creates you what is known as phosphatidyl inositol, okay? An inositol ring with a phosphatidyl molecule attached to it. Okay, but we don't want phosphatidyl inositol. We want phosphatidyl inositol 4,5-bisphosphate. So we need to add a few more phosphate groups onto this inositol ring. So, if this is, all we need to do is label up the carbons in this ring, and then we're done. Okay, so here we'll call this one that is actually attached to the phosphate group carbon 1, and then we'll just work our way around. So we'll call this 2, 3, 4 is over here, so we'll stick a phosphate group off its alcohol group, okay, and then 5's here, we'll stick a phosphate group off its alcohol group. Okay, so here we go. Here are 2 more phosphate groups, and this now is the structure of phosphatidyl inositol 4,5-bisphosphate. And I hope what I've convinced you of is that it is just a modified phospholipid. So it's a normal component of the phospholipid bilayer. All of your cells will have this molecule within their phospholipid bilayer. Okay, and really it's just a normal phospholipid with a bigger head, basically, which is sticking into the cytoplasm or into the extracellular fluid. Okay, so in the next video what we'll see is what this enzyme, PI3 kinase or phosphoinositide 3 kinase, is going to do to this molecule, but you may well be able to guess. It's a kinase, and it has even given you a number. Okay, so have a think as to what you think this is going to do to the phosphatidyl inositol 4,5-bisphosphate. You might even try naming the product that you think is going to be produced.